So hello class, today we are going to start with our topic, the introduction to bioseparation and downstream processing. So we must know that what basically is bioseparation, what basically are the downstream processing, what are the processes, and along that we also would like to know that how after the fermentation, if once the fermentation is done, how are we going to process our product to get the actual product. What is the actual product? The actual product is not only the product, the whole of the product that we have harvested from our fermentation. The actual product means the purified product in which the form in which we are going to use it, in which the consumer can utilize it. So in this, first of all, we need to know what is bioprocessing, because this part on the whole is a part of bioprocess. So what is bioprocessing? Bioprocessing simply means the utilization of living cells. How many kind of living cells do we know? The animal cells, the plant cells, and the microbial cells, or their components. What are the components? Components like their cell organelles, their enzymes, etc. for the production of utilizable products. What are the utilizable products? Like we already know that by these cells we can produce antibiotics. Antibiotics may be used for pharmaceutical purposes and other kind of drug molecules, certain kinds of organic acids, and there are very many other kinds of products that can be utilizable. So the bioprocessing literally means the utilization of living cells or their components to manufacture the new products that are for the benefit of human beings, that are benefit for the society. Next, how are we going to produce these products? By the help of the bioprocess. Bioprocess basically has three main objectives, three main disciplines. First of all is the upstream processing. What is upstream processing? We are going to discuss about it in short. Next comes the fermentation. Fermentation is already a part of upstream processing. The third one will be downstream processing, which is of our main objective in this lecture. So first of all, what is upstream processing? Upstream processing means all the processes which are utilized to rise in a power product. From the initial point, the starting point from where we are going to collect the materials which can act as either the raw materials, the media, the cells, and we are going to collect all the things and we are going to start up the process, the continuation of the process until and unless we end up with the bioconversion and we end up with the production of our product. Next will be the stages of downstream processing, where we are going to purify that product that we have produced. It means that we are going to get the product. Suppose for say that we have taken the example of our antibiotic. If once the antibiotic is produced, how are we going to purify it? So in the upstream processing, the first objective is to get the cell. Which kind of cell? The isolated pure cell that is used for the bioconversion. Bioconversion means to get production of that product which is required. Next thing, what is required? First of all, we need to collect the cell. We need to isolate it. If it is isolated in the pure form, then it is okay. Otherwise, if we are not having it right away, then we are going to isolate it from the natural habitat from where the cell is available to us. After that, we are going to check that the required product is being produced in the enormous amount which we are requiring. If not, then we may use different kinds of techniques like genetic manipulation techniques, which you already know, like genetic recombination, or using the artificial methods like recombinant DNA technology. These all genetic techniques are going to make the cell available for continuous production of our metabolite. In sense, if the product is being produced in certain circumstances only, then we are going to genetically modify it in terms that it will be capable of producing the required product in amplified amounts. Next comes, if once the cell has been isolated, then we also need to provide it some kind of nutrition so that the cell can survive, isn't it? So for that sake, we need to optimize the media sources. We need to know that what are the essential carbon sources on which that particular cell is going to survive. We need to know that what all are the extra components that are to be provided to the cells, like the growth objectives. We can say that there are very many kinds of compounds which are required for any cell to grow. Like certain cells requires particular amount of nutrients, not only nutrients, but there are some other things. These could be like the vitamins, these could be like a particular kind of amino acid. If that is not available in the media, then the cell might not be able to grow well. So that kind of nutrients are also to be provided. So in that sense, we have already maximized, optimized our nutrient media. Third thing comes, how are we going to make the cell survive in the conditions? The conditions are also very, very essential. How are we going to control the conditions? First of all, we need to understand that what are the conditions in which our cell grows optimally. Optimally means it can survive at its best. It is going to replicate fastidiously. It is going to produce our product of interest at a much higher rate. So once we have provided the media in its optimal source, secondly, if we are available with the optimal growth conditions, like we know that what would be the optimal pH, what would be the optimal temperature on which it is going to survive. The third thing comes that we can now counter check that is this cell capable of growing in the optimal conditions in this particular media which we have designed. So after that we are going to perform a trial run. Trial run in what sense? Trial run means we are going to merge all these things into a particular bioreactor which is a very low volume bioreactor. Say like the bioreactor is only of 1 liter capacity, that is usually available in our laboratories, which we call them as tabletop fermenters. In such fermenters, we are going to pour the media first, then we are going to inoculate the cells under the sterile conditions by maintaining all the sterile conditions. After that, we are going to give it a fermentation run that could be either for some hours to some days, depending on the quantity, the product which is required, and the bioconversion rate of that product. We need to analyze all those things. Once this production step has been completed, fermentation is over, now we will go on for the next step, that is we will raise up the fermentation level. How are we going to raise up the fermentation level? That now whatever was the product in our 1 liter fermenter, we are going to check it, then we will go on for the next level of fermentation, maybe say for fermenter of 100 liter capacity. Then thereafter, if the fermentation is okay in that, then we will proceed for next level, that is for 500 liter fermentation, and then at last for the industrial level fermenter. Why are we going to do that in that stepwise manner? Why not we go for directly for the commercial level fermenter? The reason is, if we are going to fail directly into that level, the industrial level, then the cost would be very much high. So why do risk for such a large amount? If we are going to just keep a check over it, we can directly check it into the laboratory level. If it fails into that, then it is much more easier to access whatever are the parameters that we are not in order. We can fix those things. But if once we are failing directly into the commercial level, that will cause a huge loss, and it is very much difficult to gain access to the basic problem, whereas it is easier to gain access to the problem in this kind of small level fermenters. So in the next slide, we are going to see that how this total process of upstream processing is being done. In the first step, we will be viewing that how we are going to access the cell that is of our interest, which we call them as the producing strain. The producing strain has been isolated, it has been subcultured, again and again its subculturing is being done. Why are we needed to subculture it again and again? So that if we have used the one batch of the cell has been utilized for fermentation, we are still having hold of that particular cell that is used, that will be required for the next cycle of fermentation. Along with it, we also need to subculture the cells because the viability of the cells is limited to up to certain generations, as we know in the majority of the cases. If we are not subculturing it, the growth of the cells will be slowed down, and at last, due to the scarcity of the nutrients and the deposition of secondary metabolites, the cell is likely to get dead. So next step after the upstream processing is the fermenter. Fermenter also is visible in the slide. You can see that first we are going to do it in the laboratory scale fermenter, then we are going to give a hike to it, and we are going to ferment it into the second stage fermenter, that could be a little higher volume fermenter, like say 100 to 250 liter fermenter. Then we are going to raise it to the third level, that is a little bit higher level of fermenter, which are usually kept as R&D level fermenters, in which it is a fermenter of say 1000 liter to 2000 liter, 2500 liter capacity. Thereafter, we are going to go for the commercial level fermenters. These could be from 1000 to several thousand liter capacity. These are the fermenters where actual production of our product is being performed. The third step will be After the fermentation is over in the commercial level fermenter, then we are going to harvest out our product. Harvesting means we are going to all the materials that are present in the fermenter, our broth along with the product that can be present either inside the cell or it could be present outside the cell, along with cells that are present in the system. We are going to harvest out all the material that had been poured into the fermenter. Now from this particular point we are going to say that we have now entered into the downstream processing. So what is the basic thin line difference between the upstream processing and the downstream processing? Till the upstream processing we were in increasing the product, we were in the phase of initiating the process till the completion of the bioconversion process, that is we call it as the fermentation process. Once the fermentation is over and we have harvested out our product, not the actual product but the product of fermenter, that will be containing what all things? It will be containing the not so purified product that will be mixed into the cells, that will be mixed along with all the other materials like the broth that are present into the system. Now we are going to purify first initially in a crude form, then slowly and steadily in our later stages, that would be in our downstream processing. So we need to understand now what basically downstream processing is, why we are calling it as downstream process. We call it as downstream process because these are the stepwise procedures in which the whole quantity of our product will be stepped down, but after each stepping down we will be having the product in much more purified form, in much more utilizable form, that is pure in its orientation sense. Now what are the downstream processing? These are the various stages of processing as its name suggests. After the completion of the fermentation, after the bioconversion of our product. Why do we need to perform the downstream processing? To get the purified product. What are the basic processes? The three basic processes underlying downstream processing are separation, purification and polishing and packaging of the product. Why? First of all, separation. Separation is the initial step which tells us how are we going to separate the unwanted materials from the required product. In separation, in the phase of separation, we might not directly go for exact purification of our product. Means that we are not going to directly purify our product into the best pure form available to us, but we are going to make a crude separation process in which the unwanted particles are separated out. Next is the purification step, which counts for very many steps. Purification means that we are now going to purify our product from the rest of the materials in the best suitable forms, in the best pure manner. The third thing will be polishing and packaging. What is polishing and packaging? Polishing and packaging means to make our product converted into that form that is in the most usable and most convenient forms. Say for example we need organic acids. So for organic acids too, I need to keep them in states like I can keep them in powdered state, I can keep them in crystallized state, or I can directly keep them in solution state. But say if I am having an antibiotic that is to be injected into a patient, so can I directly keep it in forms like powder form? Will it be more convenient? Or for injection purposes, if I keep it in the purified format in a solution state, that would be more convenient. I think that would be much more convenient. If I have to use a particular kind of source for say edible practices, like we all know that there are very many additives like citric acid, an organic acid that is to be used for edible practices. So now do I need to gain it into directly that form, that is that purity level that was required for therapeutic purposes? No, that is not the case. In this we have to just purify it up to that level, that much is required for edible purposes. But if I am using that same product for therapeutic purposes, for the medicinal purposes, then the purity level have to be very much higher. Along with it, we also need to get the product into some different kind of state. So the purification and packaging means in which particular state we need to gather the product, in which particular state the product is required. So let's discuss the various stages of downstream processing. Before discussing each step, we must understand that the total cost of the production, the total cost of starting from the collection of all the materials that is required for bioproduction till we get the final purified product, the whole lot of amount, that is we can say that more than 50% of the total production cost accounts only and only for the downstream processing. That is, it is a tedious process that is to be done, but in the same sense it is very much cost consuming. The other problem is, with each and every process, with each and every step of downstream processing, the cost will rise. Along with it, cost is also rising, and the product that was available initially will go slowly and slowly in less amount. With each step of downstream processing, the total product cost will rise, but the total product of the total volume of the required product will decrease down. So we have to keep in mind that we have to use only those steps which are essential, which will purify our product in the best possible form, and by requiring least number of steps we have to get our maximum purified product. Clear? How are we going to do it? These steps for the downstream processing includes, first of all, the removal of the insoluble material that are present inside our product that is to be purified. Next is product isolation. What is product isolation? We are going to isolate our products from the rest of the materials in the best possible method, in the best possible way, so that it is not going to disturb our further procedures. Third is product purification. These are the steps which are going to make our product purified in the best possible manner, in the best possible pure form. The last will be packaging and polishing, as we had just discussed. So now we are going to see the exact what are the stages in which our downstream processing has been divided. So our downstream processing has been divided into the separation process, which we had already discussed. Next is the disintegration. Disintegration will be required in only certain cases, that is why it has been marked with a sign which indicates that it will not be required in each and every case of downstream processing. Third will be extraction. Extraction means that we are going to extract out our product from certain remaining. Next will be concentration. In the concentration step, we are going to concentrate our product in the format so that it is present in large amounts in the total volume of the we are having. Last will be purification. Purification will be the final step in which, after which we are going to get our product in the best purified form. That will be now called as the actual product. And last after purification will be drying, if it is required. Drying simply means that we can dry our product if it is required in either the crystallite format or in the powder state. The last would be the packaging or polishing, in which we convert our product into forms that are user friendly. User friendly means it is easier to use, it is easier to store. Clear? Now let's look on to these steps. All these steps are placed in a manner such that it is easier to handle the product. It is feasible to go straight away with the nature of the product. Nature of the product means if the product is say polar, so we are going to use certain different kind of steps, and if the product is non-polar, then we are going to use certain different kinds of method. So first of all, we need to know that what is the nature of the product, and we have to assess all these kind of factors for the sake of undergoing these different kinds of downstream steps. What are these factors? These factors are size. Based on the size, that either the cell is small, we are going to use different kind of techniques. If the cell size is larger, we can use certain different kinds of techniques. Similar in that way, shape of the cell also matters, shape of that molecule also matters. Next is the density. If the product is of higher density, then we can use some other good type of techniques, but if the density of the product is very low, then we have to use different types of techniques. Similarly, the solubility will also cause different kinds of effect onto that. Next comes the diffusivity. You already know that if the product is readily diffusible with certain kinds of compounds, then it will cause ease to separate with certain techniques, but it will on the other hand may cause difficulties by separation with certain other kinds of techniques. Next is the polarity, and the same way is charge. So these are the factors which are going to cause changes in these all steps. Based on these factors, based on all these factors, we have to step down or step up in all these techniques. We can use many kinds of techniques. So in the next slide, we are going to see that how are these techniques differentiated. If once I say that using this particular step, that is disintegration, in certain cases disintegration will be required, disintegration of the cells will be required, and in some cases the disintegration will not be required. As it is available into the slide, disintegration of the cell will be required only in the case if the product is intracellular. Intracellular means the product is available inside the cell, or it should be present in both the cases, that the product is intracellular as well as extracellular also. And if the amount of product intracellular, in the case of intracellular products, if the amount of product is higher inside the cell also, that is to be harvested out. Only in those cases this disintegration will be helpful, otherwise it is not to be done. Then in the other cases we directly have to separate out this. We will be directly proceeding from separation to extraction procedures. So now let's look on to all these steps. First of all, the separation of particles. How and what basically is separation and how it is going to be done? The separation simply means that we have to separate out the two things. As we recall that from the fermenter we have taken out the harvest, which will contain broth along with the product that could be present either into the broth or inside the cells, or the second thing will be the cells. If we separate out these two things, then it would be easier for us to perform the later on all these steps. These are the following steps. If we are not going to separate these two things, then it would be much more difficult for us to separate all those things. So separation means we are going to separate the cells from the broth. There are various techniques for separation. The techniques could be used are filtration, next centrifugation, then flocculation and flotation. Let's discuss about these techniques in a short manner, because just this is our introductory lecture and we are going to revise these topics in a large format in our next lecture. So what is basically filtration? Filtration simply means, as we have already done in our life very many different kinds of filtration procedures. What is filtration? Filtration is simply that we are going to use certain kind of filter through which we pour something and the filtrate is being collected and the required product can be separated out from it. The same kind of process is being used in our industrial applications, wherein we are going to filter out our product. A thin membrane could be used, or a selectable membrane can be used, which is going to filter out any kind of thing. It is basically using the nature of either the shape and the size of that particular material that is to be filtered. There are very many problems associated with filter for which we are not going to use this technique. What are those things? Things like we have to apply any kind of pressure. Pressure means either we have to overpressure it from above or from under. How are we going to create the pressure below the system? We can create vacuum inside the chamber wherein the filtration is being done. Problems associated are, if our product is very much viscous, like if suppose you can imagine that if we are filtering something that is as viscous as honey, so can we, if we are having a simple mesh and we are going to pour honey over it, will it be easier to filter it? I don't think. It would be very, very difficult. In the same way, if somehow the filter has been clogged, clogged means the impurities have already blocked the total surface of that filter, will it be easier to filter? No. So these same problems will be associated in the industrial fermentation also. Next is centrifugation. What is centrifugation? Centrifugation is a technique which is using the centrifugal forces, the sedimentation of the particle, and we are going to use the same nature of that particular product. If the product is larger in size, if it is having higher density, then it is more susceptible to settle down. So we are using to exploit the density. If the product is of higher density, if it can settle down by applying centrifugal forces, then we can separate out such kind of products by centrifugation. So what are the problems associated with centrifugation? How many types of centrifuge do we have? And there are lot many things that are to be discussed. We will discuss this topic separately. The third is flocculation and flotation. In the meanwhile, if you want to see what is basically a centrifuge and how it looks like, so it is available in the slide. Next is flocculation and flotation. What basically is flocculation and flotation? Flocculation means simply in certain cases, if the cell is we are not able to separate the cells from the media, then in certain cases this kind of techniques could be used. In certain cases, if we are having some inorganic salts, some hydrocolloids or certain other kinds of organic polyelectrolytes, we can add this system. In certain cases, the cells along with such materials are having a capability to adhere together. These cells can stick together if certain kinds of compounds are present. This sticking together of the cell will cause one thing, that is the cells will keep on sticking together and it will cause flocks. These flocks will be of higher density, their weight would be much more higher in comparison to single suspended cells. So we call them as flocs. Now these flocs will be much more easier to separate, like in techniques that we had already named, like centrifugation, or by simple means of separation, by simple means of filtration. Later on, if this thing is also not correct, if it is not also working, then we can go for next thing, that is flotation. We usually do one thing, that in a flask that we are having, we simply pass air with much more intensity. A high intensity air, the air bubbles are passed. These air bubbles will stick to the cells and it will help the cell to rise onto the topmost layer of that particular fluid. If once this thing has been done, then we can simply harvest out the cells from the surface, or we can use other techniques for separation of these cells. Why? Because now the cells will be separated and it will form a separate layer from the other remaining fluid, that could be either the broth or some other thing. So that these are the two techniques, flotation and flocculation. We are going to discuss it again in the broad aspects, that how these things are going to be done. Next is the disintegration of cells. As we had already discussed, in which cases the disintegration will be required? It will be specifically required only and only in the cases of the product being intracellular. Means the product is present inside the cell, and to get access to that product, we first need to disintegrate the cell. We first need to break that particular cell and harvest out our product. So there are very many techniques that are available for disintegration. These could be either by mechanical lysis of the cell, means we are going to mechanically break that. Next thing will be chemical lysis, in which we are going to use certain chemicals which are capable of breaking the cells. Third will be the enzymatic lysis. There are very many enzymes available with us nowadays, which are capable of breaking the covering of the cell and gaining our access to that particular compound, that will liberate that compound outside the cell. So first of all, what is mechanical disruption? It will count for very many approaches, like it is available in the slide, that three basic mechanical cell disruption methods are: first of all, shearing. Shearing means that we are going to provide some heavy kind of shearing to the cells, which is going to make the cell disrupt. An example is grinding it in a mill. The second one is pressurizing the system. How are we going to pressurize the system? We are going to simply, as you know, that if we are going to pass something through a very fine nozzle, that will cause a very big pressure over to that cell. Due to that high pressure, the cell will burst up and it will liberate all the material out. Third will be ultrasound. Ultrasound, as you have already studied in your back courses, that ultrasound is also a good medium for breakage of the cells. So we are going to utilize this technique for the disintegration of the cell. And the important point, again I am repeating, why I am repeating it again and again, because you have to keep in mind that disintegration is not to be done in each and every case. It will be specified only in the case of intracellular products. Next kind of disintegration of cell we can do by which technique? By lysis of cells. Lysis either by chemical means or by enzymatic means. Certain chemicals are going to lyse the structure of the cell, which are going to burst the cell open. Same way, by the biological systems, like the various available enzymes, we are going to burst open the cell. These are going to lyse the cell wall and the other cell components, which will liberate out the product. Product means not that particular actual product, but the product of our interest that is present inside the cell. The next downstream method, that is extraction. What is basically extraction? Extraction counts for the process of removing of a product from a mixture of, say, the cells from the solvent phase into that particular system. The extraction will help us to achieve the two different things that we have discussed. It will help us to get separation as well as the concentration of that required product. Along with it, it is also especially useful in the cases of recovery of compounds like antibiotics and lipophilic substances, which are lipid loving, means which are having both kinds of nature. They can be either hydrophobic loving or hydrophilic loving. As you know that lipids are having the two kinds of natures. The third is extraction is also especially useful in recovery of antibiotics and lipophilic substances. Along with it, there are very many approaches that are available to us. Approaches like liquid extraction, then whole broth extraction, and the third is aqueous phase extraction. What is liquid-liquid extraction? In liquid-liquid extraction, it will employ two immiscible types of liquids. What are the two immiscible type of liquids? Which are not readily miscible into each other. Like for say an example, if we are going to mix water along with oil, yes you can say, or some other kind of non-polar compound like say chloroform or benzene, are these going to mix together? No, they are not going to mix together. In certain cases, what is to be done? They will form the two separate layers, and we can simply do one thing: the product of interest, the product that is to be harvested and to be collected into the pure form can be directly dipped into that. We are going to agitate it firmly. After the agitation, we will allow it to stand for some time. In the meanwhile, when it will stand, it will separate out into the two different phases. The first one is the polar, the second one will be non-polar phase. The product, according to its nature, if the nature of the product is polar, then it will dissolve into the polar phase, and if the product is of non-polar phase, then it will dissolve only in the non-polar phase of that particular system. Now these two systems will be separated with a marginal difference which could be even visible to us. In that manner we can directly separate these two phases. Now we know that if the product is of polar nature, then we are going to access only the polar phase, and if the product is of non-polar nature, then we are going to access to the non-polar phase only. In this sense, this liquid-liquid extraction can be done. I think this kind of experiments you might have done in your schooling, wherein we were performing those kind of experiments. If not, then we are going to proceed it into our laboratory classes. The second one is whole broth extraction. What is whole broth extraction? Whole broth extraction counts both the cells along with the media, the nutrient media that is available. So if we are going to extract it, in that case we say it as whole broth extraction method. This extraction method reduces the number of extraction steps. So it is a good technique if we are able to employ this one. In this, it reduces the product loss. Why? Because the number of steps are low. The number of purification steps are reduced. So it will reduce the production cost. Same way, however, if the effectiveness of the extraction may be reduced due to presence of cell. Why are we saying that? Because if the number of then it might risk that there are of impurities that might be present into that system. The third and the last technique of extraction is aqueous multiphase extraction. It is used for separation of enzymes from the cell debris. If we are directly harvesting out the enzyme, then we can use the system. The other point is enzymes are extracted in aqueous pegged phase. What is pegged? That is polyethylene glycol dextrans mixture, which forms the different separate phases. Similarly, by using this aqueous multiphase extraction, the enzyme recovery from these phases is comparatively very much easy if we compare it from the available techniques, from the rest available techniques. Next is the concentration phase. What are we going to do in the concentration? We are going to concentrate our product. As the name is itself suggesting, what is concentration? We are going to get our product in much more quantity in comparison to the large amounts. The total amount of the sample that is containing the product will be decreased down, but the concentration of the actual product that is present into the system will, say, suppose if the total product available to us is say 100 liter after fermentation, but the required amount will be 100 grams into that. And if after a certain procedures, if we are going to bring it down to the total, now the total product is available as say 1 liter, but now the actual product is present in how many amount? It is present in say 70 grams or 80 grams into that particular solution. So you see that the concentration of the actual product has increased, but the total fermented product is now decreased. So these are this all thing can be done by concentration. There are very many steps for concentration. First of all, evaporation. What is evaporation? You all are very much familiar with this term. Evaporation. Simply, we are going to evaporate our product by using various techniques. Evaporation is an efficient arrangement that must be made for recovery of evaporates to reduce the cost. Next, it is used in cases of solvent extraction. The various devices which could be used for evaporation are, say, continuous flow evaporators, falling film evaporators, thin film evaporators, centrifugal film evaporators, as well as spray dryers. We are going to study each of these differently in our whenever we are going to discuss it into our succeeding lectures. Next are the membrane filters. What are membrane filters? These are simply the membranes which are used for the filtration of substances. So a little bit of information is also available into these slides, and we are going to discuss it into our next lectures. Next is ion exchange reagents. What are ion exchange reagents? For understanding it, we need to recall our memory for the what are ions and how are these going to help us in the concentration of our product. These are basically the polymers which are firmly attached to an ionizable groups which ionize under a suitable environment. How these polymers are going to adhere this product that is of our interest on the basis of its ionic nature, or we can say that on the basis of their charges present over them. Either these reagents could be either in the solid form, say the dextrans, cellulose and polyamines etc. can be used as ion exchange reagents in the solid format. Otherwise, if we require liquid, then we can use phosphoric acid or mono or diesters. The next are the adsorption reagents. These are similar kinds of reagents, but these are not going to use the charge. Instead, they are going to use the sorbity, that is the adsorption capacity of that particular substance. So these are the porous polymers without ionization, which are capable of adsorbing the material to itself. Their porosity determines the surface available for adsorption. Means the more porous this kind of reagents are, the more will be the adsorption. And these reagents may be either polar, semi-polar, or they can be non-polar. These will be used as per the nature of the particular product of our interest. Next, we will be talking about the purification, which is considered to be the last step where the purified product has to be achieved. What is to be done into this purification step? There are actually a group of techniques which we now commonly call it as chromatography. Chromatography is the best known technique for the purification purposes, and it is classified under purification. Why? Because after chromatography or after this purification step, we are directly accessible to the best purified form of our required actual product. So let's look into this. This slide says that it is the final step in the product recovery which aims at obtaining product in the highly purified form. It is the highly purified form that would be available after the completion of this purification step. How are we going to do it? We have the two simple methods. First is the crystallization, the other one is chromatography. Crystallization will be done in those cases wherein high purified product is not required. Say, as we have already discussed the example of citric acid, if it is used, it is to be used only for the edible purpose. Like we sprinkle the citric acid over our fruits so that they do not get oxidized. So for such practices, the highly purified format will not be required. Whereas if the citric acid is to be used for therapeutic practice, then it has to be highly purified, and for that purpose we are not going to crystallize it directly. We are going to perform which technique? We are going to perform chromatography. Chromatography itself has very many diverse techniques into it, and we may use according to the nature of the product that which technique is to be employed when. So crystallization, let's talk a bit about it. Crystallization is mainly used for purification of low molecular weight compounds like antibiotics. Crystallization is the final step in purification of products like citric acid, sodium glutamate and antibiotics like penicillin G, etc. Whereas chromatographic methods, that is the major counterpart of our interest, it says that these are used for purification of low molecular weight compounds for mixtures of similar molecules, like for say example the homologous antibiotics which are somewhat having similarity in them, which are very difficult to separate, like the various other kinds of penicillin antibiotics. You know, there are very many kinds of antibiotics there are very many forms of penicillin. So if they are very slightly different in their structures, so these can be differentiated on the basis of by using the technique of chromatography. Next, the different kinds of chromatographic techniques, I am just going to give out the names. We will discuss them in our next classes. These are adsorption chromatography, affinity chromatography, ion exchange chromatography, covalent kind of wherein we will be using the interactions for the sake of chromatographic separation. Third will be gel filtration, and at last the partition chromatography. So these are the major techniques which are of our concern. Next step which I told you, after purification, if it is required to be done, then we may undergo for it, that is drying. What is drying? Drying is simply to convert our liquid product into the non-liquid states, let's say into the crystals or into the powdered form. So it could be done either by vacuum drying, by spray drying, or simply by freeze drying. The drying is required because it makes products suitable for handling and storage. Say if the product of our interest was, say, the whole cell, like in the case of brewer's yeast, it is used for bakery industry and for various other purposes. So if we need to store that particular cell, then we have to dry it in some format. Drying not directly by heating it, because it will cause the cell to, that will simply kill the cell. But we are going to dry it in some other manner so that the cell viability is also not lost and we are able to store it for quite longer durations. There are very many techniques that I had already told you. These are vacuum drying, spray drying and freeze drying. We will be discussing them later on. Last will be dialysis. Dialysis is also used only in certain cases. That is why we are calling it as an extensive approach, as it is visible to you in these slides. These are extensive approach because this could be used in the fermenter itself if an option is available to us that the product can be filtered out by the means of dialysis. Dialysis, you people are already having some kind of knowledge about it. In dialysis, we simply provide a semi-permeable kind of membrane through which the product of our interest can be separated. So keep that in mind, that how the actual dialysis was done. I am telling you that how dialysis is done in the fermenter. If the fermenter may contain a chamber separated by a selectively permeable membrane, which allows the product diffusion from fermenter chamber to dialysis chamber. So there are two different chambers available. Next, in this way the product is recovered while the fermentation continues. In the fermenter, the product is also being purified and separated side by side. The fermentation is also proceeding. So the fermentation along with the separation is going continuously. Next is, this approach can be used for the removal of lactic acid during fermentation by lactobacillus species. So this has been particularly used in industrial implication of the production of lactic acid. Next, the product polishing. Product polishing simply means that we are going to finalize the product in a stepwise manner so that at the end we are going to get the product into the desired form, that will provide ease of handling and ease of storage to us. What are the steps that are done in the product polishing? We can either crystallize it, we can desiccate it, we can lyophilize it, or we can simply spray dry it to get the product in our utilizable format. So this is all that we have to discuss into our introductory class for the bioseparation and downstream processing. I hope that everything would be clear to majority of you, and from our next class we will be emphasizing on that. First of all, what are the niches that are required, then we will be proceeding on to our technologies. That is all.